Alright, today our next tutorial actually is going to follow up heavily on Gordon's ligand protein docking. We're actually going to do protein docking and give a real world example that you can follow along in your tutorial. Protein docking is a later, sort of more complex algorithm that was added later to Rosetta. Rosetta's initial features were, of course, for de novo folding, but protein docking takes upon the knowledge-based potentials that Rosetta uses and allows them to expand to more complicated applications like protein docking. So the overview is just we're going to go over why do we do protein docking and how exactly the Rosetta Monte Carlo grid search works. And then I'm going to try to explain the difference between a low-resolution docking procedure and also a high-resolution docking procedure. And then our real-world example will be an actual antibody-antigen docking example with some data analysis. And then finally I'll show you some references and resources where some servers, namely the Gray Lab, have taken upon themselves to automate some of these processes. Not as robust as what I'll be showing you here today, but automated nonetheless. So why do we do protein docking? And I've got this quote from Jeff Gray, who is the one who initially wrote the protein docking algorithm for Rosetta, and he says the post-genomic goal is the characterization of the structures of protein complexes, and computational tools offer an inexpensive means to carry out large-scale studies. Basically, all he's saying that everything that happens in biology is a protein interaction, pretty much, and the large time and expense to characterize each protein interaction could be streamlined with a computational tool, and that's exactly what the Rosetta protein docking algorithm does. So, speaking to this crowd, I don't really need to go over what protein interactions, how important and vital they are biology, but they're involved in such processes: protein localization, competitive inhibition, allosteric regulation, gene signaling and regulation, the vast signaling pathways you see on any given lecture, and the signal transduction that follows. And then, of course, what I'm most interested in and what we'll be going over is pathogen clearance, antibody-antigen interactions. And in this case, I've shown that to actually turn on a T cell, we need protein interactions and peptide protein interactions, and this sort of stuff is done with Rosetta dock all the time. So we can characterize how proteins interact in two ways, and we know principles from chemistry, and this is more of a molecular mechanics approach where we give it some sort of real-world potential and have it interact that way. But everyone knows the fallbacks of computational chemistry, where we're trying to do large complexes. The system gets too complex, and so we borrow from Rosetta the knowledge-based potentials, where the information from the PDB and laboratory experiments and thermodynamic measurements fit directly into the Rosetta algorithm, and it limits our search space, that infinite search space that you see in de novo folding and pretty much all aspects of computational biology. So the protein docking works exactly like you think. If we consider these two puzzle pieces, we want them to know how they fit together, and so we fit them together by translating them and rotating them. If these are viewed as one rigid body and another, what's the lowest energy conformation that will allow these to bind? And we want to know if they bind, how they bind, and what effects do mutations have on binding. All can be done with Rosetta dock and protein docking. So the old way is where we discretize the receptor and ligand, and we just turn it into a crude grid search space. This is not how Rosetta works, but this is the older methods before Rosetta came along. And we just want to know, how does this shape fit into this shape, and when they overlap to give the best interiors touching the exteriors, the score is reduced, and that is your complex. There's some inherent problems with that. Is because it doesn't take into consideration all the little interactions that happen on the amino acid scale. So Rosetta does incorporate the amino acid interactions that are allowed to move and rebuild and repack upon protein-protein docking, so it gives you a better answer to what the solution is. So Rosetta uses a Monte Carlo search of docking space coupled with full atom repacking of side chains. What I mean is we just search along the protein using the other protein, and once we find it, we put in side chains and see if the interactions, how exactly interact at an atomic detail. In ensemble docking, we do this Monte Carlo search plus the side chain repack, which is the new part added to Rosetta, as I mentioned. And we can also account for the small backbone perturbations. To view two proteins as rigid bodies upon docking is probably not how it happens most of the time in nature. So this ensemble docking can account for some of the smaller backbone perturbations. Today in the example we won't be doing ensemble docking, but we'll just be showing two rigid body orientations. So the Rosetta dock is a Monte Carlo with minimization steps, and it cycles in between the rigid body translations and then repack minimize steps. So I know these have been gone over, but sidechain optimization is just a search through the Dunbrack libraries, and then the rigid body minimization is just a gradient-based minimization. So you can see here we start, we move our protein somewhere along the space, we do a repack which will lower our energy, and then a gradient-based minimization, and this should be our final structure. It may be a local minima, it may be a global minima. That's why we show that Rosetta needs to be ran many, many times to search this entire energy landscape. So that's important. So this is just the Rosetta dock algorithm that it was initially published in this gray paper, the resource I've given at the end. We have a random start position, and then we do a low resolution search and a high resolution search, and I'll talk about that. And the low resolution decoy, once we find it, we repack those side chains like we talked about. We do a minimization step, and then do we accept it? Yes or no. If not, we move it again, and this is how we traverse our energy landscape. So the difference between a low resolution and high resolution step. The low resolution step turns the side chains into essentially centroids, which are representations of the rotamer, and fits them together. It allows faster sampling of the conformational space. And the high resolution actually puts all the atoms back in and then does smaller perturbations on the protein to actually get the final structure. These do not necessarily have to be coupled together. If you want a rough, if you're perhaps doing epitope mapping, you want a rough search of where the epitope is, all you'd probably need is low resolution Monte Carlo search. But if you want to know to atomic detail how the proteins interact, high resolution refinement is needed. So in this filter, these parameters are given from experimental data. So for instance, you could know that some part of the protein doesn't interact at all. So you could say, if it's bound at that part of the protein, I reject it because I know biophysically from my experiments that this interaction does not occur, and we can reject it as follows. So that's how Rosetta really couples well with experimental energy, because we can filter out anything we don't know, or anything we do know at Chase. So this is how we're going to use protein docking. So for this example, we're going to assume we know nothing about the orientation of either binding partners, and we're going to do a global dock that includes both a low resolution search and high resolution refinement. So the MDM4 protein is just some random protein I pulled from the PDB, and it comes along with a single IgG chain. And this is the native structure here, so it's tightly packed at the interface. For instance, we do know that antibodies will always try to bind at their antigen binding sites, but we're going to assume for this case we don't know that. So all I've done here, and if you follow along in your example, I've taken the single IgG chain and moved it out, moved it out 10 angstroms or so, and then we're going to see if we can find this native pose again. So in the PDB preparation, this is important. So this is actually two chains in the PDB called PDB chains B and C, and this is chain H. For this to be viewed as one entity, we have to change it so the chain IDs will be the same. So we take B and C and label as chain B. This doesn't hurt, since Rosetta just works at, it terminates after it sees the chain, and then it can view this as one entity and this as one entity. So it's very important that we compare our PDB in such a way. That way it knows exactly what proteins to dock. If you did not do this, it would try to dock, it would keep this rigid, and it would try to dock C to B, which we don't want. We know that's the native complex. So if you follow along, we are using Rosetta scripts, and Rosetta scripts is Gordon talked about, and if you it allows for modular movers. So if this hasn't been explained, a mover is anything we actually do to the protein, such as minimization, repacking, docking. Those are all movers, but they are defined in protocols such as docking. Now, if we want to change anything in the protocol, we'd have to hard code it in. Like if we wanted to do an extra amount of minimization steps, we'd have to hard code it in. But if we use the modular mover, we can simulate the Rosetta docking protocol, but we can do it in Rosetta scripts, and Rosetta scripts just means the modular way of putting together movers to simulate a protocol. And if it's not in Rosetta scripts, your application you'd like to be using, I bet it will be soon. So this is our command line. We just call on Rosetta scripts, and then I've given you the flags file that we'll look at, and the database, and then the parser protocol, which is the XML file that contains the Rosetta scripts command. So we'll look at both of those right now. So the flags files, most of these you've seen before. The parser protocol we'll call low-res docking. It really doesn't matter what you name it. This calls on the XML script that actually has all the movers in it. And then we have some docking commands that aren't specified in Rosetta scripts, but we can specify them outside in the command line to tell Rosetta scripts to do some actual other stuff. So I'm just going to go over the important ones. The docking, this specifies the degrees of freedom of rotation and translation, so how one of the monomers is able to move. This docking randomize is the flag that if we truly don't know anything about the given binding site, we just randomize the orientation to start with and then start docking them. So that we'll use this because we're assuming we don't know anything about anything about these proteins. The rest of these are just how many cycles of Monte Carlo searches you'd like to go through. Instruct, you've heard of, and then our rotamer library we run on right. And then pack and repack only is another important flag. This tells us we don't want to do design, so that's an important one to keep in there. So this is the XML file I was talking about. It has 4 movers. Dock low, where we don't do any high resolution refinement, and dock high, where we do high resolution refinement. We also have a repack move and a gradient-based minimization move. And this is these are really well documented on the Rosetta Scripts website, how these work. And then in the protocol section, we just put them together. First we want to do a dock low, then we want to repack minimize, dock high, repack minimize, and this simulates perfectly how Rosetta Scripts can recapitulate the docking protocol by modular movers. So a couple important things. The initialize from command line tag right here, this task of operations, allows us to give Rosetta Scripts some extra info. For instance, since these aren't part of Rosetta Scripts yet, you can tell Rosetta Scripts to look into your flags file for extra things to do to your protein. So the low resolution docking motor converts to centroid mode. I've already gone over this. Dock high is the refinement. The repacks the complex from the rotamers, and minimize